Good afternoon, everybody. I'm Marco Pitino. I'm a research molecular biologist at USDA in Wapato, and my research focuses exclusively on X disease. As we many talked this morning, X disease is caused by a phloem-limited bacteria and is uneven distributed in the plant. You can see here on the left top some symptoms. In a different branch, you don't see any symptoms. So it's very difficult to identify symptoms, also because they are only visible during ripening. So what is happening here? The bacteria, as you can see on the right section, is a phloem-limited bacteria, and once is there, start to producing effector proteins. These effector proteins are small and soluble. They can move far away from the bacteria, and they can target different plant proteins. And by doing so, the bacteria is shutting down the plant defense responses. So how do we target this bacteria? We have two different systems to deliver a molecule in the phloem. And one of these is called DPI, direct plant infusion. And this is 3D printed, and you can see the ring is made of silicone. This to prevent leakage. And what we do is to add any molecule in the container, and then because the transpiration of the plants is uptake the liquid in the tank. And this is an example of a young cherry tree. This is after 15-20 minutes. The blue dyes were moving already into the leaves, flowers. Then we dissect under the delivery, and the blue color was also moving down. We also did something with leaf hopper, and we add the leaf hopper on the leaves on the plants with the red dye. And 24 hours later, we dissect the leaf hopper, and you can see the eggs are red color, the gut is red color, but also all the insects show the coloration. So this could be a screening tool to test molecules, not only for the phytoplasma, but also for the insect vector. The second cell delivery system is called Symbiont, and we are taking advantage of a bacteria that is present in nature and has the ability to transfer DNA to plant genome. And by doing so, we are trying to make a biofactory on the size of the plants, and we also can include any gene of interest. So while we are growing this biofactory, the cells are also producing molecules. And because this structure is highly vascularized, we are producing and delivering the molecule into the plant. This is an example. Once it's inoculated, we are growing this structure, the product is produced, and the product can move inside the plant. This is an example of a symbiont. Far left, in the middle is a section. We cut the symbiont, and you can see the GFP expressed in the symbiont. On the far right, we have a longitudinal section. These are tomato plants. So you can see the symbiont with the core production of GFP, and some potentially leaking of GFP outside the symbiont. So the idea is to produce this symbiont on cherry and test different antimicrobial peptides against phytoplasma. So what happened? As I said, the bacteria, when it's in the phloem, is secreting effectors. These effectors are moving and targeting plant proteins, and so we have a disease. The idea is to produce molecule by the symbiont that can bind this effector, so the effector cannot bind the target, and so prevent the disease. So these are a parallel between a conventional treatment versus symbiont. So usually with conventional treatment, you need to have first a production somewhere, then you need transportation, and you have a storage. You also need to have a formulation. You are not even in the field yet. And then you have a multiple treatment. And this add cost, at every step, cost to the molecule. With the symbiont, we think that 2,000 liters, 520 gallons can treat 1 million acres, 150 million trees. And usually you do the symbiont inoculation. You have the symbiont development. The product is done by the symbiont, which then will move into the plant. And thank you for all the collaborators and the sponsor. Thank you.